One of our priorities that was identified early on is that there was a need to have a better standardized method for measuring insulin secretion.
This was an opportunity to help sharpen our tools for development of new drugs for this disease.
The first goal was to standardize the two methods that we had selected, the arginine stimulation test and the mixed meal tolerance test with minimal modeling. The second goal was to measure the variability or reproducibility of these two measures across the glucose tolerance spectrum.
So the output of the Beta Cell project is a standardized method that allows our tool to be used in large clinical outpatient trials. Being able to measure how the beta cell function changes over time allows development of a therapeutic.
The advantage now is that through this project, we have really characterized how these different measurements of beta cell function perform.
The Beta Cell project team intentionally had members from both the public sector and the private sector.
This whole notion of bringing the stakeholders together to work and solve the problems commonly, people then own it. They own it together, and that collective is critical.
So the Beta Cell project knew going in that the goal was to make data available to all so that all academic researchers and any other interested parties could have access to the standardized methodology and the known characterized variability.
The more we standardize, the more we can compare across studies and the more we can pool data in order to get a better understanding of the questions we're trying to answer. Ultimately, this is about the patient and what we can do to understand this disease better and develop better drugs for these patients.